Good morning, ma'am.
Oh, good morning, Pranava. Slides are they visible?
Yes, ma'am.
Yes, ma'am.
Okay.
Good morning, ma'am.
Good morning, Lekhya.
Good morning, ma'am.
Good morning, Ma. So I think people are joining, right?
Yes, ma'am.
Yeah, yeah. You can go ahead. I am there. I've got connected. So we will see. Yes, right?
Okay, ma'am. Sure.
Yeah, the children have come, all of them.
They are still joining, ma'am.
Okay, okay, Ma. Right. Thank you.
Thank you.
Good morning, ma'am.
Good morning. Good morning, Ma. Good morning, everybody.
Morning.
Good
Morning,
morning.
ma'am.
Good morning. Pragna, Chinmayi, and Pranava. Right. Good. Good to see all of you. So in a short while, we'll all join together. Thank you.
Vijay Bharat? Pragna, am I audible, Pragna?
Yes, ma'am.
Ah, Bharat, could you please check how many joined in the YouTube?
Actually, your voice is echoing, ma'am.
Am I not clear, Pragna?
Ma'am, you're clear, ma'am.
Is my voice okay?
Yes, ma'am.
Bharat,
Yes, ma'am.
how many of them joined in YouTube, Bharat?
YouTube, ma'am. Wow, I will check, ma'am. Madam, 13 members, ma'am. 1 3.
Okay, I request all of you to post a Zoom ID and password to all your groups. First year, second year, and final year, both sessions. I request all student volunteers to post ID and password to all our groups.
Okay,
Am I audible?
Yes, ma'am. You can.
ma'am.
Good morning, Principal Ma'am. Good morning, Vijay sir, Manohar sir, Sridevi ma'am. Pragna, am I
Good morning,
audible?
ma'am. Good morning. I am here. Yeah. But there is some little disturbance coming, Lekhya. Little bit of sound disturbance is there. Otherwise, everything is fine.
Sound, ma'am.
Ma'am,
Yeah.
actually,
Some
your
sound
voice is giving
disturbance
an
is
echo,
there.
ma'am.
My voice?
No, Lekhya Priya, ma'am,
No,
your
no.
voice is
I
giving
think
an echo.
Lekhya's voice. Yeah, it is getting.
Ma'am.
Yeah, some sound disturbance is there.
Yes, ma'am.
Pragna, could you switch on your video, Pragna?
Yes, ma'am.
Now am I clear, Pragna? My voice?
No, ma'am.
Echo is coming, ma'am. Lekhya,
Yes,
echo
ma'am.
is coming.
Okay, ma'am.
Just check, ma'am. In your voice, the echo is coming actually.
Okay, ma'am.
Yeah. I think there are two devices side by side. That's the reason why there is some echo, ma'am. Otherwise, it will be fine.
Yes, ma'am.
If you have kept any other device very close, you can switch it off.
Okay, ma'am. Now is it
Yeah.
okay, ma'am?
Yeah. I think still the disturbance is there. If any other device is there, you can keep it at a distance.
Okay, ma'am.
Yeah.
Now is it okay, ma'am?
Yeah, it's fine, but there is some blurring voice coming like here.
Now, ma'am, am I? Is it
No,
okay,
the
ma'am?
same. No, ma, it is the same thing. You're using mobile side by side, or somebody else is using. Anybody? Yeah.
I have muted, ma'am.
Yeah. Okay.
Is it okay now, Pragna?
Yes, ma'am.
Is there any disturbance from my side?
Now once again it is coming, ma.
Ma'am, actually I have switched off everything, ma'am.
Maybe some other device is there connected there with the children. You can check. It need not be yours, like it can be anybody's.
Oh, okay, ma'am. Ma'am, now is it clear, ma'am?
There is the clarity is there, like but there is some blurring voice, blurring sound is there, disturbing sound.
Now, ma'am, is it okay, ma'am?
Yeah, I think so, ma. It is fine.
Maybe earphones.
Yeah, I think now it is fine. Yeah.
Okay. Thank you, ma'am. Ma'am, most of the students have joined. Shall we start, ma'am?
See how many participants are there, ma? Here we have 28. That means
Yes.
around 25 students.
Yes.
Then on YouTube,
Bharat and Mahesh.
you know how many people have joined on YouTube, ma? Here we have around 25. And exactly it's going to be 11 o'clock. So YouTube, can you just check how many people have joined?
Bharat,
It's about,
tell me. How many joined in the YouTube student volunteers?
13, ma'am, on YouTube also.
30.
On YouTube, 13, ma'am.
Okay.
13, 1 3.
Shekhar has joined, Lekhya.
Yes, ma'am. Sir has joined.
He has joined. Okay. So exactly 11 o'clock now. Shall we wait for 5 more minutes or shall we start?
Ma'am, according to your wish, ma'am, no problem, ma'am.
No. I'm just asking, ma'am. We are all ready. So what shall we do? If you want me to start, then we will straight away start. Otherwise, we need to wait for 5 more minutes or start. Shall we then?
Okay, ma'am. We'll wait for 5 minutes and we'll start, ma'am.
Okay, we'll wait for 5 minutes, and you need not wait for my my concern. You can start, ma'am. 11:05 exactly. We will start.
Okay, ma'am. Thank you, ma'am. Dear student volunteers,
Yes, ma'am.
post
Yes,
YouTube
ma'am.
live stream link also.
I did, ma'am. I did, ma'am. I posted.
Yes, ma'am.
Ma'am.
Pregnant.
Yes,
Now
ma'am.
my voice, is it echoing?
Yes, ma'am. Now it's echoing. Before that, it's not echoing, ma'am.
Now pregnant?
Yeah, it's fine, ma'am.
Okay, fine.
Shall we start, Lekhya?
Yes, ma'am.
Yeah.
Over to you, Pragna.
Shall I start, ma'am?
Yes.
Yeah, please start.
Yeah, ma'am. Honorable Chairman of Sri Vidyaniketan Educational Institutions, Dr. M. Mohan Babu, sir, our dynamic revered CEO, Mr. Vishnu Manchu sir, the principal of Sri Vidyaniketan Degree College, Dr. D. Ashalata ma'am, teachers and delegates of today's program. A very delightful morning to one and all. I am Pragna Chinmayi of second year BSc Microbiology, Sri Vidyaniketan Degree College, Tirupati. On behalf of Department of Biological Sciences, I feel privileged to welcome you all for the international webinar on biosynthesis of metallic and metal oxide nanoparticles and its nanomedical applications. Now, I humbly request our respected principal ma'am, Dr. D. Ashalata ma'am, to give welcome address. Please, ma'am.
Yes. Thank you, Ma. Thank you very much. It is indeed a great pleasure that today, once again, we are in a whole lot of excitement as we are into the series of once again conducting the webinars. And today, it's very importantly, like, the active kind of topic that we have in the world today, the nanotechnology related topic. And this is an international webinar that we are hosting today on the biosynthesis of metallic and metal oxide nanoparticles and their nano biomedical applications. So we are indeed very happy and very honored that Dr. Jacob Vijay Kumar, who is a postdoctoral scientist from Nanyang University, China, has accepted our invitation and agreed to be part of this program and being the resource person of this program. We are indeed very thankful to our management and because our honorable chairperson Padma Shri Dr. Mohan Babu sir and our very revered CEO Sri Vishnu sir always encourage us to conduct these kind of activities because they pave avenues for the exploration of research of both the faculty and the students. Indeed, it's a great kind of an opportunity when we actually think about what exactly is required in this world to make the children more actively participate in the global conservation activities. So when it comes to the nanotechnology, of it we have been seeing that there's a lot of medical exposure, and in the past 10, 20 years, we have seen that there's a lot of green synthesis which was happening everywhere, whether it is about reliability or sustainability or trying to go for the environmental conservation. Everywhere it was about like how we have to reduce the effects of the phytochemicals or the toxicity which was everywhere prevalent in all sorts of areas and the applications which are happening in every different fields of science and technology. But then, when it came down to the green synthesis, it was very active that the plant-based or the natural ingredients were used for the biosynthesis of metallic oxides. Because, say for example, in our culture, we have often seen, whether it is in India or the West, we have often seen that the metals are used as utensils or they are used for different purposes or accessories in our life. So we actually eat from the metals or we wear the metals and it is part of our culture. So sometimes these metals are used just to have a kind of an soothing effect or a therapeutic effect on our body as well. But then normally when we take about the metals like gold or silver, copper or zinc, which are part of the system of the culture or our traditions or even for daily day-to-day traditional life. But then when the oncological kind of targets were given to the nanotechnology, or maybe it was very much used in the medical biotechnology or whatever it is, what has happened is that there was more of the effects of this radiation or the metal oxides which were used in a different way because maybe intravenous or maybe ocular or the dermal kinds of effects that it had, or sometimes it has affected the other parts, maybe sometimes smell or maybe the brain. The metal oxide components, when they were not degraded or they were not synthesized properly, they had severe health effects on the body. So it was really understandable that this has to be dealt with, and in order to do that, the biosynthesis was very much required. So as a nanotechnology part of it was explored or it was going on, a lot of research has paved way or it has given a kind of scope for the researchers to understand that biomedical properties have to be explored and the nanoparticles have to be applied in the way of the nano medical applications, because when it comes to antimicrobial agents of the metallic oxides, or because they are used for the DNA imaging or the labeling or the molecular synthesis, or when it comes to the kind of metals like palladium or selenium also are widely used in the medical labs or in the medical therapeutic usage. It is necessary that they have to be treated properly before they are being used for medical applications. So I think this is a very what we call as a emerging area of research which has to be explored. And I hope that the students and the faculty who have joined us today, the program we are conducting on behalf of the Department of Biological Sciences at Vidyanikethan Degree College, we are very glad and we are very happy that this is going to be an interesting topic for the students to explore. And we are very much thankful once again to Dr. Shekhar Vijay Kumar for accepting our invitation and for being here with us today. I hope that whatever effects that we have with the nanoparticle usage of the metallic oxides, when they are treated properly, they are going to have excellent antifungal or antimicrobial or anti-cancer, whatever, whatnot, lot of implications that we have in the medical field. I think they are going to improve the quality of life and reduction of pain as such, which is very important for each and every one of us. We hope that the world will become a better place with researchers like Dr. Shekhar Vijay Kumar who are working in these kind of areas which are required for actually dealing with the pain and suffering that the human beings are going now, as we have less of vaccines and more of viruses and diseases. Thank you so much, everybody, for joining us today for this international webinar. And I once again congratulate the student team, the coordinating team who have done wonderfully well to gather such a big crowd today and publicize the event. And I also congratulate the department and the coordinating faculty members for doing so well and continuing this webinar series. So God will abundantly bless all of us to take up much more activity and very good research topics in the coming future. Thank you one and all, and God bless all of us.
Thank you, ma'am. Now I request our UG and PG coordinator, Dr. P. Manohar sir, to share his thoughts on today's webinar.
Thank you, Pragna.
Thank you,
Respected
sir.
Principal Madam, Dr. D. Ashalata Garu, Webinar Coordinator, today's Webinar Coordinator, Dr. C. Lekhya Priya Madam, Organizing Members, Head Department of Biological Sciences, Ravikant Sir, Hriday Madam, and Technical Support, Head of the Departments, other Head of the Departments, Faculty Members of SVDC, and all participants and student volunteers, especially today's resource person, Dr. Shekhar Vijay Kumar Sir, PDF China. A very good morning to all. This is Dr. P. Manohar, working as a UG and PG coordinator of Sri Vidyanikethan Degree College. It's my privilege to welcome you all for this huge gathering of this international webinar on biosynthesis of metallics and metal oxide nanoparticles and its nanomedical applications. It's a good topic. We are enthusiastically waiting for listening these words from Vijay Kumar sir. I appreciate all the participants who are highly responded and registered to this great webinar. And first of all, I congratulate all the student volunteers who have been given their service to our Vidyanikethan Degree College. I am sure that this webinar will help a lot in a great extent to all the participants. I am confidently saying that this webinar will goes to reach our expectations of all the participants. And about the webinar, last few years, there has been a growing interest in using nanoparticles in different biomedical applications such as targeted drug delivery, bioimaging, and biosensor, etc., etc. As a recent survey says that around 150 companies across the world, they are working on nanoparticles in drug therapy to treat wide range of diseases such as fungal and bacterial infections. The pace and demand in any field, be it genetics or nanotechnology or cosmology, can only met with increased international cooperation and collaborative projects. Definitely, this would help a lot for all the participants. Finally, I congratulate Dr. C. Lekhya Priya Madam and the organizers of this international webinar, which is essential in this situation, being every human being are interesting to understand about nanoparticles, because in future, there is a lot of demand for nanoparticles and nano equipment materials because of this growing industry. And I congratulate Dr. C. Lekhya Priya Madam and organizing members for conducting this great seminar, international webinar. I conclude my this one, and once again I thank you one and all for giving me this opportunity. Thank you very much. Thank you,
Thank
ma.
you, sir.
Okay.
Sir, please continue.
Thank you, ma. Thank you. So
Okay,
please.
sir. Thank you, sir. I request our webinar coordinator, Dr. Charles Lekhya Priya, ma'am, Assistant Professor, to introduce today's eminent resource person. Over to you, ma'am.
Thank you, Pragna. A very good morning to one and all. It gives me great pleasure to introduce and welcome the resource person of today's international webinar, Dr. Shekhar Vijay Kumar. Dr. Vijay Kumar sir received Doctor of Philosophy in Biosynthesized Metallic and Metal Oxide Nanoparticles, Potential Applications and Ecotoxicity Assessment from Alagappa University, Karaikudi, India. Dr. Vijay Kumar Shekhar is currently working as postdoctoral research fellow at Marine College, Shandong University, China. Dr. Vijay Kumar is doing research in nanopharmacology and nanotoxicology, nanobiotechnology, biomaterials, biofilms, infectious diseases, ecotoxicity, limnology, immunology, and zoology. He has published more than 40 research findings in topmost peer reviewed index journals with a total impact factor of above 100. Dr. Vijay Kumar is a reviewer of the international journals like Elsevier, RSC, and Spring journals, and also the editorial member of Environmental Microbiology Journal. He has participated and presented research findings in various national and international conferences. Dr. Vijay Kumar has sound knowledge in the concepts of physico-chemical characterization, analysis of nanoparticles, MALDI-TOF, RT-PCR, green fluorescent protein-tagged bacteria cultures and maintenance, freshwater and marine fish live feed culture and maintenance, species identification and maintenance, and its use in the isolation of biomaterials, biopolymers, immunological, toxicological, and host-pathogen interaction studies, mosquito cultures and maintenance, species identification, control of mosquito vectors using nanobiomaterials and bioactive compounds. Other skills include molecular docking, molecular simulations, and dynamic blast pasta expasy tools, phylogenetic tree construction, analysis of DNA sequences using vector NTI software usage, and other bioinformatics tools. Dr. Vijay Kumar was an invited resource person and delivered 9 special lectures. He has membership in professional bodies such as Indian Science Congress Association, Association of Microbiologists of India, and Indian Biophysical Society. Dr. Vijay Kumar received several awards and grants. Some of them are University First Rank Holder in MSc Zoology, DST Inspire Fellowship, Outstanding Reviewer Award from various elsewhere journals, Top Reviewers in Pharmacology, Toxicology, and Crossfield, September 2019 Publon. Sir, despite your busy schedule, you have accepted to be the resource person, and on behalf of the organizing committee, I thank you and welcome you for today's session. Thank you, sir.
Thank you, ma'am. I deem it an honor to invite today's esteemed resource person, Dr. Shekhar Vijay Kumar sir, to enlighten us about biosynthesis of metallic and metal oxide nanoparticles and its nanomedical applications. All the participants are eagerly waiting for you, sir.
Yes. Okay. Thank you, ma'am. Good morning to all. First, I would like
Good
thank
morning,
to.
Dr. Shekhar.
Yes, ma'am. Good
Thanks so
morning.
much.
Yeah, good morning, ma'am.
Thank you. Thank you so much. God bless you.
Ma'am, can you see my PPT?
Yeah, sir. It is there. You can do the presentation, sir.
Okay. Thank you, ma'am.
Thank you. Thank you.
Can you see my PPT?
Yeah.
Yeah. Okay. Good morning to all. First, I would like thank to principal ma'am and head of the department and organizing secretary and students. Thanks for the opportunity to my present to my research and share my research. I hope It is very useful for the UG and PG students. Okay, I'm going to talk in my today topic biosynthesis, metallic and metal oxide nanoparticle and nanomedical application. Before going to my talk, I'm just tell about my institute and research. I'm working as a postdoctoral fellow in Shantung University, Vigai, China. This China in Vigai city is a very good city. It is a very cool place, and we are working in Marine College. We are working in the biomaterial preparation of the biological sources to test it for the some cancers and tissue engineering application. We have prepared for the some marine sources, some seaweeds or sponges to take samples and to analysis the preparation, isolate the compounds to prepare the nanomaterial and drug delivery application, some tissue engineering application for the biomaterials. Um, this is topic. I'm going to talk in biosynthesis of metallic and metal oxide nanoparticle and nanomedical application. What is mean? It's a biosynthesis. It is used for synthesis or preparation of nanoparticle in the biological resources, as was plant or microbials or biopolymers, or it is the biological sources, renewable sources, or non-renewable sources to obtain from synthesis of the nanomaterials and to tested the medical applications for the using the biosynthesis nanomaterial. For any queries related research collaborations or any queries regarding the topic, if any time feel free to contact me going through the link on my mail and my mobile number. Okay, then what is the nanotechnology? Nano, first it is in the sense the nano. It is a Greek word. It is a very in small size. It is a dwarf. It is the range. It is the 9 to 10 to the power of minus 9 in nanometer. It is the nano range. It is a 10 to the power of minus 6 in the micrometer. This particle is very tiny or very small range. It is a nanoparticle in nanosite. In the first term in the nano technology in first introduced in the concept in 1959. The Nobel laureate Richard Feynman was the first introduced into the nanotechnology in the meeting of American Physical Society. Uh, he said there is plenty of room at the bottom. They first introduced in the concept of nanotechnology in 1959, Richard Feynman. Afterwards, in 1974, the term nanotechnology was coined by Professor Norio Taniguchi. After in the 1974, after the year in 2000, in US approved for the nanotechnology and nanotechnological application in the various projects for the US in nanotechnological applications. So what is nanoparticle is a unique physical and chemical properties because compared to the bulk counterparts, it is a very unique properties because the nanomaterial is a highly surface area. It is a very important factor of the nanomaterial. The nanomaterial application, the various versatile usage for industry, textile, medical, mechanics, haptics, for the application for the nanotechnology. Basically, the nanotechnology, the nanoparticle approaches for the synthesis in the two-way for the nanoparticles. One is the top to bottom approach. Another one is bottom to approach. First one, top to bottom approach, it is the method etching, laser ablation, milling, rending, sputtering. It is the method for the chemical methods and physical methods, chemical and physical methods used for the top to bottom. It is the what is the method is the main factor in the top to bottom approach is the bulk material converted into the small particle to the top to bottom approach method. It is the method. It's a very disadvantages like us chemical methods used for the toxic chemicals, environmentally harmful. That is the main reason. Another one is a physical method used for the we need for the lot of spaces and high pressures and highly manpower used for the physical methods. This is the top to bottom approach method. Another one method, bottom to approach. It is the very easily friendly method. It is used for the synthesis of biological synthesis used for the bottom to approach method. It's a bottom to approach method used for the it is the small part bulk material converted into the small or nanoparticle into the atoms nucleation process because it's a bottom to approach method. It is a subdivision in the Sol-Gel process and laser pyrolysis and chemical reduction, spray pyrolysis, and biological process. It is the bottom to approach method used for the some capping or reducing agent used for the bottom to approach method, like plant extract or microbial extract or any biological sources extract or capping for the nanomaterial preparation to the bottom to approach method. It's a biological process. What is the biological process used for the nanomaterial or nanoparticle synthesis, like plant, yeast, bacteria, algae, and fungi? These are the capping agent for the preparation of the biological process of the nanoparticles. It is a method, I'm already previous slide explained, top to down process. Bulk material convert to the fine particle, it is a top down process. And another one, bottom up process. The bottom up process, it is a material convert to the bulk material, convert to the small nanoparticle. It is the involvement of atoms and nuclei. It is a convert to the nanoparticles. Basically, nanoparticle classified into the two categories. One is organic nanoparticle and another one inorganic nanoparticle. Like organic nanoparticle include carbon materials, it is a carbon sources. Another one, inorganic nanomaterial, like metal and metal oxide, like silver, gold, zinc oxide, titanium dioxide, silicon dioxide. This is the inorganic nanomaterial. In generally, basically, the inorganic nanoparticle in past 10 years, the research is very highly peaked to the nanomaterial preparation and used for the various application process. And type of materials in nanoparticles, like inorganic nanoparticle, metallic nanoparticle. First one is a gold nanoparticle. Gold nanoparticle, which is one of metallic nanoparticle. It is used for the last previous more than 2000 years. It is a Leucurcus cup. It is a globlet in colored using gold nanoparticles. It is a red color and another one orange color. It is a 20 nanometer and 40 nanometers. Previous 2000 years ago, it is the gold nanoparticle to identify. And gold nanoparticle, it's widely used for recently, current scenario, gold nanoparticle used for various application, particularly in biomedical field, because high electron conductivity and minimally low toxicity and very biocompatibility for the drug delivery applications for the gold nanoparticle gold nanoparticle usage. Because only disadvantages, it is highly cost in compared to the silver, and almost it is a very biocompatible good candidate for the drug delivery preparation and drug combination preparation to the application used for the various cancer drug delivery and other applications. Another one, palladium nanoparticle. Palladium nanoparticle is one of the metallic nanoparticle. It is the catalytic reaction. It is a very good catalytic property. It is used for the palladium nanoparticles environmental application for the removal of environmental degradation dyes, various textile and industrial dyes removed for the palladium nanoparticles. And silver, the silver is everywhere. Silver is easily it is not high cost, it is affordable, and silver nanoparticle used for the highly preference because the highly antibacterial and antifungal property for the silver nanoparticle. It is also inhibited the various biofilm formation also. Already in current scenario, some silver nanoparticles already quoted in already available in the commercially available in the market in silver nanoparticles. Another one, copper nanoparticle, is a copper. It is one of the metallic nanoparticle. It is used for the some of the good catalytic property and also the good antifungal property. The copper also, it is used for the various agricultural purposes because the plant enhancer and the plant reducing the microbial infections, also the prepare the copper nanoparticle, and water remediation purpose used for the copper nanoparticle to used for the industries. And another one is inorganic nanoparticles and metal oxide nanoparticles. Metal oxide is a zinc oxide and titanium dioxide. It is a very majorly used for the various application purpose. Zinc oxide, one of the metal oxide. It is a non-toxicity. It's a main advantage. It is the very low cost and the non-toxicity and very good biocompatibility. And it is GRAS as generally recognized as safe. It is approved from FDA, Food and Drug Administration USA, approved for the GRAS certificate. because zinc oxide nanoparticle, it is almost we day by day increasing the some cosmetics items also. We used to for the some lacto elements, some h god ointments, also some ingredients of the zinc oxide, some ingredients in cream formulations and zinc oxide. It is a highly antibacterial and antifungal activity also. Another one, titanium dioxide nanoparticle. Titanium dioxide is also used for the some cosmetics ingredients. It is a very UV protection agent. Zinc oxide also, because it is a preparation for the some cosmetic formulation to prepare the some titanium ingredients used for the preparation. It is a highly antibacterial and antifungal agents. Then after synthesis, the nanoparticle, it is a very important to identify the nanoparticle, which is it is nanoparticle or not, to tested or identify the some characterization used for the nanomaterial. First one UV, another one XRD, FTIR, SEM, TEM, EDX, zeta potential, TGA, BAT. First one UV, each and every nanoparticle, it is a different character and different wavelength and a different shape and size. This is a physicochemical characterization. UV, it is a UV for the to tested or identify the nanoparticle in the optical property, to tested the nanoparticle. Example like silver in 400 to 500 nanometer range in surface plasmon resonance SPR rate. In example 400 to 450 in the range of silver nanoparticle, and 300 to 400 in the range of zinc oxide nanoparticles, like optical property, to tested the identify the optical property in UV ultraviolet spectroscopy. And another one, XRD, X-ray diffraction. The X-ray diffraction meter to analysis the goniometer to analysis the crystalline nature of to identify the nanomaterials. To XRD, it is the phase, what is the lattice to present in the XRD to identify the nanoparticle samples. Another one, FTR, Fourier transform infrared. It is the instrument, it is the KBR, potassium bromide in nanoparticle mixed in the method used for in the what is the biological function, what is the biofunctional groups present in the sample to analysis the FTR analysis. Another one, SEM, it is a surface morphology analysis of to identify the nanoparticle scanning electron microscope. Another one, transmission electron microscope. It is used for to analysis the size and shape. What is the size? What is the size range to analysis the transmission electron microscopy. It is a highly advantage field of the microscopic techniques to tested for the nanoparticles. Another one, EDX, elemental composition and purity to tested the EDX analysis. And the zeta potential to tested the what is the surface charge and what is the stability to identify the determine the nanoparticle to test the zeta potential analysis. TGA, thermogravity analysis to test the what is the temperature degradations in the material to purpose of TGA analysis. Finally, one PET. PET is one of the important instrument analysis to test the nanomaterial surface analysis. What is the surface area? In the Brunet-Teller, it is analysis. Then biogenic nanoparticle, inorganic nanoparticle, biosynthesis, it is the biosynthesis, I'm already previously explained, in biological synthesis, the lot of advantages compared to the chemical and the physical methods. To preparation of the researchers is moving on in the biogenesis or biological synthesis of nanomaterials. It is a very less expensive and non-toxicity. It is a biocompatibility is a main important factor of the biogenesis synthesis of nanoparticles. It is various applications to used for them, some metal oxide and the metallic oxide, metal nanoparticle used for the various application to the nanotechnology. The nanotechnology interdisciplinary science is the physics, chemistry, and biology. It is everywhere in the nanomedicals and nanophysics and nanochemistry in various in the interdisciplinary area, nano, and recently in the nanomedicine also. It is the method, different methods of nanoparticles synthesis. I'm already first slide explained. It's a plant material, biological sources. Example, plant extract to plant extract metal salt reduction to after the synthesis into the identify the color observation and the other material characterization, like plant extract to the add silver metal or gold or zinc oxide material. Another one used for the bacteria. Example, in probiotic bacteria to intracellular or extracellular culture to tested the probiotic bacteria with the example like zinc oxide nanoparticle to tested to application for the aquaculture industries for the probiotic nano feed for the nanoparticles. Another one, fungi, fungal also fungi hyphae used for the growth media and prepare the salt solution to prepare the nanoparticle. Why the researcher going, it is move on the biological source like plant or bacteria, sorry, fungi used for. It is because the plant and the fungi are the present in the various phytochemicals or phytocompound present in the extracts. It is some secondary metabolites also. So it is the very biological properties to enhance the very good antioxidant activity and also the various biological application. It's a various mode of action. It is a nanoparticle to kill the disturb the this see the figure disturb the bacterial membrane and what's the mechanism interactions with the DNA RNA damage the ROS generations. It's a plant sources used for the metallic nanoparticle in the previously researchers extract from different plant pots like flower, fruits, stem, fork, seed. It is used for the preparation of the nanoparticles in various nanoparticle synthesis like silver, gold, or zinc oxide. Then important of phytochemical plant extract the biogenic preparation of nanoparticle. These are the phytochemicals in the present in the plant compounds or some fungi compounds or secondary metabolites like polylaids, glycosides, alkaloids, carbohydrates, polyphenols, polysaccharides, ascorbic acid, vitamin C, flavonoids. It is the phytochemicals compounds present in the plant extract or fungi to advantage of preparation for the nanoparticles. This advantage of nanoparticle. Why? What is advantage? Very good advantage. It is the nature of solvent. It goes easily preparation, simple procedure, not used for the toxic compounds, and high reducing potential and less contamination, and environment not environmental impact. These are the some chemical structure of the chemical constituents or phytochemical constituents of and responsible for bioreduction of silver nitrate in the silver nanoparticle previously reported in the nanoparticles. So one is a compound affine, ascorbic acid, and mufol, and the tannic acid is tannic acid, tannin. Tannic acid also present in the tea leaves, Camellia sinensis, then the pincombrine, caloge, and pilanthin, gallic acid, and some of the eugenol in the glove compound, and the allicin, it is used for the garlic, it is some of the compound. This is a protocol for the phytosynthesis of nanoparticle to preparation of plant extract and the mixing the metallic solutions. And after the synthesis, mixing the solution, the bioreduction mechanism of the metallic and metal nanoparticles. And after the nanoparticle synthesis to tested the various physicochemical characterization, UV, XRD, SEM, then we have to analysis the once confirmation of the nanoparticle size and shape, all the physicochemical characterization, move on to the biological applications. In the noble metal nanoparticle using plants because various some of the compounds, root, latex, seed, it is used for the nanoparticle preparation in the various plant sources. Another one used for the some plant extract and microbial extract used for preparation of nanoparticles. Another one important aspects in biopolymers. Biopolymers, it is the source from its animal source or plant source, and the synthetic polymers. Um, Another one, natural polymers. The biopolymers, it is the method to the biologically sources softline. It is used for the some preparation of various biomaterial and drug delivery application, and used for the preparation of nanomaterials. What is the advantage? It is the biopolymers. It is a very biodegradability and very good biocompatibility for the preparation of a nanoparticle. The biopolymers, it is also used for the bioplastics. These are the biopolymers. One is a polysaccharide, plant and algal origin. Some of the biopolymers name starch, amylose, amylopectin, cellulose, agar, alginate, fucoid, and carrageenan. It is some seaweed polysaccharide biopolymers. And animal, chitin and chitosan. It is derived from the animal sources. It is present in the crab and shrimp cells, cells, outer shells. It is obtained from the chitin, chitosan, and some hyaluronic acid. And bacterial origin, some dextran, gellan, leaven, gums. It is also used for the bacterial biopolymers. Another protein, the collagen protein, gelatin, and gluten. It is also some synthetic polymers, polyesters, polyhydroxyalkonates, and the specialty polymer, natural rubber, and synthetic polymers also preparation for the nanomaterials. In ranking nano patents and nano article in Russia in 2020, patents for 100 article, last update in June 2020. In the first upgrade in the USA, 51.74% in the preparation and production patents of the nano used for the nanotechnology in the applications. In the application, the degree by the ratio in the maximum 51.1 for major in USA and the 9.7 in fact in UK. Then move on to the therapeutic applications, biological applications, sorry, nanomedical application used for the phyto or biosynthesis of nanomaterials. It's a biomedical application of nanoparticle in the various purposes like drug delivery, therapeutic material, and the drug interaction, and the preparation of new drug and the preventive strategy infectious diseases also. The nano, what is the biosynthesis methods in compared to the highly preferred surface area and the highly chemical and physical properties to the some of the nanomaterial. Example like nanomaterial recently preparation the some cancer drug loaded preparation like aptamer, tendramer, and liposomes preparations for the nanomaterials recently developed in the imaging concentration and the bioconjugate nanoparticle to the drug delivery application in the various dreadful diseases, and also to used for the nanomaterial to the environmental application, degrade the dyes and the drugs. This is nanoparticle products in already available in the commercially available in the market like bandage, silver coated, wound healing or wound bandage, and another one silver coated socks. It is antibacterial agent because it is a gill bacteria, silver coated socks, and the silver sanitizer like silver ointment, the healing for the wound healing and antifungal agents. And another in the USA, some other countries, it is used for the water purifiers, although the silver coated water purifiers and sun cream lotion like zinc oxide and titanium dioxide, and some of the wears, inner wears, although the napkins, women napkins, and the dresses, jeans like also the silver coated and the zinc oxide coated, the nano products to preparation for the nanotechnology. And the cancer drugs, new drugs in the combination preparation for the anti-cancer drugs preparing for the nanomaterial. Already some of the India also the preparation of the silver coated Samsung washing machine available for the in nanotechnology productions. In Thailand, it is some of the paint wall paintings in available for the nano paints. It is used for the antibacterial, for the nano paints used for the commercial available in the market, and hair gels and some t-shirts also. This is and another one important factor in the Hummer nano composite parts preparation for the Hummer cars and the various badmintons and the creams and the mobile factors and the hygienic inner software solution used also for the nanotechnology in the market. Then let's move on to the biological or nanomedical application. What is the application to the nanomaterial? But first one, it is the antibacterial activity of nanoparticle. The nanotechnology, it is a nanoparticle like metallic or metal oxide or carbon or whatever it is, it hydrogel, like whatever it is, first initiate to the going to the tested for the antibacterial to tested the nanomaterial. Day by day increasing the novel contamination or infectious disease for bacteria or virus. The bacteria day by day increasing the resistance, more resistant to the commercially available drugs, because we have alternatively needed for the alternative vaccine or drug needed for the bacterial or viral infections. So the tested for the nanomaterial to antibacterial activity. In my research, I'm preparing for the nanomaterial. Laurel lophilis, it is one of the plant. It is commonly known as bay leaves. It's biryani leaves used to preparation for the zinc oxide nanoparticle to tested the clinically important pathogen. One is Streptococcus aureus and another one Pseudomonas aeruginosa. Both are the clinically important bacterial pathogens. It is also forming the biofilms. We have tested, you see the image T1, T2, T3. T1, T2, T3, it is a treatment, different concentration. The concentration increases, the zone of inhibition level also increases. It is a zone, it is formed with the inhibition. It is a bacteria culture. The C means it is a control. Another one, Pseudomonas. Pseudomonas culture also the C control in the various concentration, highly inhibited bacteria. This paper I published in the Biomedicine and Pharmacotherapy. If you have any reference, go through it. Another one, anti-biofilm activity of nanoparticles. What is the biofilm? The biofilm, it is the bacteria, a group of bacteria. It is a formation, cluster-like formation, not a single bacteria. It is it is more than 1,000 times fold multiplication of the single bacteria, like it is a biofilm bacteria. The biofilm, it is the mechanism of cell-to-cell communication, like man-to-man or human-to-human talk. Like it is a forum sensing mechanism. The biofilm is more resistant to the antibiotics because it is control. It is the eradicate or control is very difficult. The more the biofilm, it is a present of bacteria in some medical devices and the catheter urinary tract and some of the medical devices present in the biofilm forming bacteria and also the present the biofilms. The biofilm also in the present in the not clinically environmental and also is a some marine environment. It is some boatings or corrosions for accumulation water pipelines present in the biofilm bacteria also. It is eradication process is very difficult to the now the researchers is controlling of the biofilms to tested the nanomaterials. Like see the it's one of my research. It is a like one, and first one is the methicillin resistant Staphylococcus aureus bacteria. Below the images Pseudomonas aeruginosa. It is above images is light microscope images, and below the images is a confocal laser scanning microscope images. Confocal laser scanning microscope, it is one of the recently advanced microscope. It is used for the look for the live cell imaging for the confocal laser scanning analysis. You see, it is a control. It is a clumpy cluster-like formation. After the treatment, different concentration, the bacteria adherence or cluster-like formation reduce the thickness and reduce the colony formation to the inhibition of the nanoparticle in highly concentration. You see, control is a higher formation, and after the 100 microgram treatment, reduce the bacteria and the colony reduced in the above light microscope, below the confocal microscope. It is treated. This is one of the 3D analysis of the biofilm analysis in the treatment of MRS areas and Pseudomonas aeruginosa. 3D analysis of the confocal laser scanning microscope analysis. It's a control, highly thickness, and the treatment it is a reduce the thickness. You see the arrow mark to reduce the thickness and reduce the bacteria. To tested the synthesis nanoparticle, silver nanoparticle like garlic extract. Garlic means glove. Garlic glove extract, right? It is synthesis for the silver nanoparticle to inhibit the biofilm activity. This paper published Photochemistry and Photobiology Journal. Right. Then anti-angiogenesis activity nanoparticle. What is anti-angiogenesis? It is angiogenesis. It is the growth of blood vessel from the existing vasculature. It is the one of the mechanism anti-angiogenesis of nanoparticle. It is the mechanism of anti-cancer activity. Further, it is the inhibition of further growth of tissue or cells in the factor to inhibit the anti-angiogenesis factor, and the nanoparticle tested the nanoparticle to the in the cancer biology studies. You see, if one of the researchers recently, I'm also working on in this assay, but not a plant because I'm also working the biopolymer. In this research, Vimal Raj et al. In 2018 reported some gold nanoparticle preparation of Mangifera indica, mango seed extract based synthesis gold nanoparticle to tested the anti-angiogenesis activity used for the chick embryo assay, chick egg, CAM assay, chorionic allantoinic membrane assay to test it is control. It's a various vasculature. It is the various veins is formation after the treatment to inhibit the already existing in the blood vessel new blood vessel formation inhibit. It is the angiogenesis effect. It is a very anti-angiogenesis effect and anti-cancer effect also. It is inhibit the further tissue formations. Then antioxidant activity of nanoparticle. That's a very important factor. We have already the Some of the free radical is more production. It is a various disease and also the formation of one important factor of disease. So the antioxidant activity of nanoparticle evaluated, it is very important one. It is ROS production, it's a defense. It is a why it is a nanoparticle to the biosynthesis used for the antioxidant activity, because already some of the food we have eat into the nano, some green vegetables also the antioxidant property, like the some of the present in the phytochemical compounds, it is the very good antioxidant property, like green tea, camellia sinensis, some of the green chilies and onions, garlic. It is also very good antioxidant property. Like this nanoparticle preparation of the various phytochemicals or phytoallergical sources, preparation for the nanomaterials in the antioxidant activity. This antioxidant, the free radical production to various disease important factor for the production of the various diseases to the antioxidant activity of nanoparticle to inhibit the disease of some other diseases. It's a mechanism, stable molecule of antioxidant mechanism, free radical production, some of the nanoparticles to used for the antioxidant properties. Then anti-inflammatory. Then what is anti-inflammatory? It is the any inflammation of the part of the swelling, redness, pain. It is an injury or some of the trauma or trauma like. It is used for the some anti-inflammatory activity of researcher recently working of the nanoparticle to tested the commercial drug like some aciclovir or diclofenac. It is an anti-inflammatory drug already commercially available in the market. It is also prescription of the doctor. It is any you have body the pain or swelling inflammation affect doctor prescription the drug anti diclofenac or aciclovir. It is compared to the drug or the recently researcher the drug coated preparation of the silver or zinc oxide coated nanoparticle to the in vitro and in vivo level to test it for the anti-inflammation or anti-angiogenesis or antibacterial activities. The mechanism, anti-inflammation, it's an injury, harmful things enter in the body, and pain, warmth, and redness in the inflammation of the process. It is like some cancer also, rheumatoid arthritis, various diseases. Another one, anti-diabetic. Anti-diabetic activity of nanoparticle. Diabetic, it is one of the important factor of the disease. It is like cancers and disease. More of the out of 10, more than, sorry, out of 10, it's 8 pupils it suffer from the diabetics. Diabetic neuropathy, diabetic nephropathy, diabetic retinopathy. Like it is already commercially available in the market, some drug, but the drugs are also do the some effects, some side effects, and also the some cells damage, vomiting, some of the various side effects also in the drugs. So alternatively, we prepare the nanos or nanosomes or nanoliposomes or nanomaterial, nanometallic coated preparation of the diabetic drugs in the preparation in vitro and in vivo level. In vitro level, like alpha amylase, alpha glucosidase, some DPP assay. Another one, the in vivo level to tested the animals in the rat, mice, or rabbit to tested the anti-diabetic levels in wound also. This is mechanism in the get in the anti-diabetic mechanism, the pancreas gut, and also the metformin. It is a drug for the already commercially available for the treatment of the diabetic. Some of the recently researched metformin coated gold nanoparticle to treat it for the diabetic therapy. Then anti-cancer activity. Cancer activity, like also the cancer is a very dreadful disease. It is uncontrolled growth of tissues and the formation. The cancer already some of the drugs also available. Some clatrin, cisplatin, some doxorubicin also the commercially available in the market. But the drugs also the some various side effects kill the healthier cells also, because the surgery and radiotherapy also in the treatment, it is a treatment is the very side effect also the damage to the normal cells. So alternative in the therapy of the nanomedicine, in the nanotherapy for the in cancer disease, in the used for the optimer and trendrimer, some gold nanoparticle also used for the in vitro and in vivo levels. It is one of my study. You see the slide. It is a human A549 lung cancer cells. Use the first one saline. It is a control, DMSO medium control. Another one is a gold chloride. It is a bare gold chloride. It's another one peel extract. I am preparing the gold nanoparticle used for the banana peel extract. Prepare for the higher concentration. You inhibit the cells. The cell morphological changes like round hectic formations and also the reducing the cell viability. It is a very good anti-cancer activity of the gold nanoparticle preparation of the use of paradisiaca banana peel extract. It is a paper published for the Microbial Pathogenesis Journal. You go any reference, go through it. Another one, and wound healing activity of nanoparticle. Wound care management, it's a very important factor because wound healing, it is a very complication process because we not correctly maintain the bacteria invade the into enter into the wounds. It is a formation of the infections more and severe complication to the healing effects in the healing delay of the process for the wound. So the wound healing activity of nanoparticle, it's a very important one in the in vitro and in vivo level. Uh, Already commercially available in the market in the some silver coated ointments. We have also used for the some wounds for the silver coated and silver sulfone aside for used for the ointments for the wound. But now researchers some biopolymer like chitosan, it is one of the very good healing agent for the wound. It's a coated like silver and chitosan coated silver nanomaterial, and I am also preparing for the some of one of the biomaterial to prepare the to prevent the wound infection and heal the very good healing activity of the nanoparticle in the wound healing infections. You see the first in vitro level, it is a in vitro means in the test lab level. It is a cell. You mean it is a wound healing activity like control. It is a like wound scratches. It's a control, not the cell migrated. It is only wound infected wound. Another one is a curcumin, another curcumin coated silicon nanoparticle. It's a, this images in the after 2 or 6 hours, the cell will be migrated. It is because the present in the curcumin and the curcumin silicon nanoparticle because it is a migratory effect. It is a healing of the wound. It is a in vitro level. It is a in vitro activity of cell migration as in the wound healing activity. Another one in vivo level, some of the researcher used for some park in Efalgar is used for the copper nanoparticle to tested the anti-wound healing activity. Like a control, it is an open wound, and after the treatment, the wound will be closed in the tissue and vasculature and collagen deposition formation also. B, it is images of the histopathological analysis for the analysis of the wound healing analysis in the in vitro, sorry, in vivo levels. Then vector control, mosquito application, mosquito vector, mosquitocidal activity of nanoparticle. In the day by day increasing the vectors more resistant into the already some chemical compounds. It's fully kill the mosquito. It is just depressed some of the compressors of the compounds in the mosquito. So we alternatively in the some researcher in recently and current research is do on the nanoparticles. So the nano vector control for mosquito side to preparation for the vector control. We have also used for the some plant extract coated silver nanoparticle preparation for the control Aedes aegypti mosquito vectors like a control images Aedes aegypti larvae, normal larvae after bare silver nitrate. It is a deposit of the silver nitrate to inhibit the body cells and damage the DNA. After the nanoparticle, it's a tiny particle. It is enter into the body. It is a guild applications and the deposition of the lungs and the heart. Deposition of the alimentary canal and deposition of the eyes. It is kill the replication of larvae further process. It is a paper published on Journal of Cluster Science. If any reference go through on it in the journal paper. Now another one, anti-parasitic activity of nanoparticle. Now the parasitic, it is a various in the parasites in the animal sources and some man-human human parasitic also. In the researcher, in my lab, one of my colleague also the preparation of the nanoparticle to prepare the pediculous human capabilities. At the human pediculous activity of in the nanoparticle preparations in the nanoparticle titanium dioxide coated nanoparticle to treat the human lice. The treatment, it's a control, it's a normal like adult, and after the treatment higher concentration the parasite will be the damages the body and reducing the pupae and larval and egg formation to inhibit the nanomaterials. So these are the like some recently medical, basically nanomedical application, some R&D parasitic mosquitocidal like application. It is also the various application also research is going on. Like drug loaded nanoparticle to deliver the stimuli responsive into the particular cancer cells and the vector controls and the diabetic control, it moves. Another one, it is the nanoparticle, not only the nanomedical application, it for the food packaging applications, and on the bioplastic productions and the food packagings and food coveration and environmental application, remove the dyes and the drugs, or the drug usage, removal drug used for the nanometal applications. Thank you. And one thing, the students after the UG and PG completed, in zoology student or biology student, what is the advantage in the field? Because it's move you to on after the MSc zoology or BSc zoology or biology, move on next to PhD level. You have to the MSc in university first rank. You have got the DST Inspire Fellowship to obtain the fellowship in the per monthly JRF level 28,000 and SRF level 30,000 rupees. You got it, and after the continue with your research or any of university in providing the some funding, JRF and SRF level and various entrance exam also available. You prepare it. All the best and thank you.
Over to Pragna. Thank you, sir. Over to
Thank you, ma'am.
Pragna.
Thank you so much, sir, for sharing your valuable knowledge and giving us the right directive to understand about the basics and recent updates of nanoparticles and nanomedical applications. Thank you, sir. Now I request Dr. V. Sridevi ma'am to begin the question and answer session. Over to you, ma'am. Sridevi ma'am.
Madam, please unmute and talk.
Okay. Thank you, Chinmayi.
Thank you, ma'am.
Good afternoon, sir. It was an excellent presentation. From a few questions from the participants, sir.
Yes.
First question: how nanoparticles used to make batteries? Batteries. How the nanoparticle used to make batteries?
um, because uh, because this battery uses some of the nickel nanoparticle and the some of the cadmium nanoparticle that is used for the battery preparation. It is the because
Yeah.
some of the powder preparation it is used for the radiate some car radiators and the batteries also preparation the solar energy to convert to the solar energy after the preparation and used for the batteries in some solar energies and thermometrical analysis.
Okay, sir. Next question. From different synthesis, different type of synthesis of nanoparticle, which synthesis is best? For example, chemical synthesis, biological synthesis, like that. Which type of synthesis is best?
More a lot of methods, but is the best method is the biosynthesis method is best. It is the phyto or biopolymer capping agent used for the preparation method. It is the best method because this environmental friendly and the non-toxic agent is used. It is very good used for the eco-friendly and the cost-effective. Cost-effective method is a biosynthesis method is best compared to the physical.
Okay, when we choose the biosynthesis method, sir, but we can't produce bulk. When we can use the chemical synthesis, we can produce bulk synthesis of nanoparticles.
Sorry, you repeated the question.
Sir, for example, we are go for the biosynthesis of nanoparticles. We are not going to produce huge amount of nanoparticles. We can produce only little quantities. Yes or no? Compared to the chemical synthesis. In the market, also chemical synthesis is more involved in synthesis of chemical nanoparticles.
Yes, ma'am. But now researchers is to move on biosynthesis because the chemical synthesis it is a very toxic because the biosynthesis process some plant process it is the plant process it is the easy process. Some other process like particular phyto compound to isolate the process is very difficult. So the researcher is move on to the some bulk quantity because it is a very good method for it is synthesis in biosynthesis because the chemical method it is not safe. So the researcher is
Sir, nowadays so much of the scientists are interested in nanoparticle research, sir. after synthesis of nanoparticle. So is there any protocol for the disposal of nanoparticles? So it should not be affect to the environment like that.
Yes, ma'am. It is you preparation. It is depend upon your selecting for the capping agent. Like you particularly, it is a plant compound. It is like example, zinc oxide. It is not safe. It is very biocompatibility. It is not harmful in the environment. In the nanoparticle preparation, depend upon the you choose the capping agent and metallic solution.
Okay. For example, lead, like that, if you use, sir, in that type, it may affect to the environment.
Yes, ma'am. Definitely, you can use in the lead. It is a very toxic. It is maybe used for in future in nano bombs and nano wars also. I think some nano agents, nano biowar agents also. Some toxic substance used.
Sir, at present, nanoparticle devices available in the market.
Hello.
Can I repeat, sir?
Can you
at
repeat
present, is there any nano? Yeah. At present, is there any nanoparticle devices is available in the market?
Yes, ma'am. In not in India, but in abroad countries, some foreign countries also there some nano devices, nano chips also present in the like video games and the some of the mobiles used for the nano chips.
Okay. Okay. Thank you, sir. Sir, one more question, sir, from the
Yes.
Ravikant, sir. Is there any device? Is there any danger to lungs and heart to the researchers who are involved in the nanoparticle research? Can I repeat, sir?
Yes, ma'am. I can't hear you.
Is there any danger to the lungs and the heart to the researcher who are involved in the research?
What is any nanoparticle toxicity, right?
Yes, sir. The person who are
Yes, ma'am. Because we have do the research in we have the well equipped and protect in process in research into the research. And we have tested the any toxicity profile. We have previous in commercially go to the market, and before we have analysis in the in vitro and in vivo level, right? Like in cell lines, and after the in vivo rat and mice, and go to the predict the toxicity level and go to the next move on to the commercial to the market. So this analysis of the if any possibilities available, like in inhale the in particle, very tiny particle like 10 to within 5 to 10 nanometer, it is very easily inhaled into the toxicity in chemical in nanoparticles. But
so that means the researchers can get the toxicity, sir, while doing the research.
No, ma'am. It is we have the well equipped and protected, not toxicity, not inhaled.
Okay.
But unprotected, powerful because maybe chance to toxic.
Okay. Thank you, sir. Sir, one more question from the student Anjali.
Yes, ma'am.
How can nanotechnology can be used in the healing genetic disorders like sickle cell anemia, like that? How can nanotechnology can be used in healing the genetic disorders? Genetic disorders.
Healing genetic disorder.
Genetic disorders.
Hello.
Yes, sir. Genetic disorder, Alzheimer's, sickle cell anemia, like that.
Like yes, ma'am. Already one of my friend also in the Alzheimer's disease in USA. He also, but not working in the nano particle, but he is the neighbor lab working in the nanomaterial for the Alzheimer's disease in the in vivo levels. Maybe in future with the clinical trials.
Okay.
In the genetic disorder, also the some populations in the animal models. That we treated the nanoparticles to the which generation to inhibit the or toxic or hereditary some gene isolations to tested the protein levels to identify the bioinformatics levels of the which genes to involved in the mutations in the nanomaterial to treated after the control and treatment levels.
Okay, thank you, sir. Now it's discussion is open to the participants. You can directly speak to the resource person.
Dear participants, you could unmute yourself and could directly talk to the resource person and clarify your doubts. Manasa, Tejesh, Pushpalata, Swati, Vaishnavi, all of you.
Hello, sir. May I speak, sir?
Thank you, thank you. Tell me.
Yes, sir. Good afternoon, sir. My name is Tejesh Kumar from second year MIC. Sir, my doubt is,
Hello,
sir,
I
can
can't
we
hear
use
you.
NAN? Sir, am I audible, sir?
Yes, yes. Now is it okay?
Yes, sir. Sir, can we use nanobots as an antibodies or antiviral agents to serve the patients who are suffering from impaired immune system, sir? It's my doubt.
Sorry, sorry. Can you repeat your question, please?
Yes, yes, yes. Sir, can we use nanobots as an antibodies or an antiviral agents to
Hmm.
serve the patients who are suffering from impaired immune system, sir?
Yes, yes. It's a nice question. Already in some research is going on to the nano antibody production, but still not accurately in the clinical trials. Maybe in future, the nano
What
antibody and
about
vaccine
yes,
productions.
sir. So what about the usage of these nanobots and detoxifying agents like can it will be used to filter the blood and removing the bacteria in our blood cells like that, sir?
Yeah, it is a preparation of the like some metals in the preparation. It is a very biocompatibility metal, like in some selenium or zinc oxide used for maybe in future for the nano antibody production because it is GRAS and very good biocompatibility and non-toxicity. So the production
Yes,
may be future.
yes. Thank you, sir.
Yes, yes.
That's it.
Sir, may I speak, sir?
Thank you, ma.
Good afternoon, sir. This is
Good
Raichan,
afternoon.
second
Yes.
year BSc microbiology.
Okay.
So you said no, sir. Which chemical is present in bay leaves, sir? Once you say, sir.
Yeah, some in bay leaves present in some linazole essential oil compounds. That leaves it's common used to for the some biryani, like biryani, some bay
Yes,
leaves.
yes, sir. Yes,
Yeah,
sir.
it is the leaves. It is present in the some various phytochemical compounds. It majorly present in the compounds in linazole, some essential oils compound.
Sir,
So
for what purpose that essential oils can be used, sir?
yeah, maybe in research in various in the essential oil used to for the various purposes like antibacterial and antifungal agent. It is used for the antioxidant like olive oil. Olive oil.
Yes, sir.
That's the purpose also essential oil. It is the one of the remedies agent for the disease control. Some of the various food ingredient agent also used for the essential oil in
Yes,
the
sir.
bakeries and some of the food product and in the recently research it's moved on to the medical biology to the essential oil to the applications in the field in vitro and in vivo levels.
Okay, sir.
What is the application of nanotechnology in tissue engineering?
Yes, tissue engineering application.
Can
It
we
is
expect a good cell growth?
Yes, yes, sir. Because in recently in our lab in my Shantang University, in our lab also preparation for the marine biomaterial, preparation for the cells, marine alternative marine chips. In the heart or lung chips to tested the biomaterial and the implants to the bone regeneration implants also developed for the alternative implants for the nanomaterial to the bone implants to preparation for the nanomaterial like titanium and like zinc. It is used for the implants, alternative implants preparation for the nanomaterials in the bone plates and joining bones plates preparation for the tissue engineering applications.
Because people who undergo dialysis, instead of going for a kidney replacement, if we go for some kind of tissue engineering and restore the damage, or I mean we can uh, heal that, that would be a huge asset.
Yes, sir, but I don't know any idea about the kidney dialysis. But the only we have now working on the implants, alternative implants for the infectious bones and remove for the alternative plates or bones in the regeneration in the some metals, calcium ions coated the marine drugs to reimplants for the patients to test the nanomaterial like some antibacterial coated plates like selenium, zinc or titanium coated. Majorly titanium and zinc also used for the implants because it is a very good effect for the bone and tissue formation, like bacteria, inhibiting bacteria purposes.
Sir, one more question.
Yes, sir.
Is nano research very costly?
No, sir, no. But all are thinking it is costly. But is the thing is the nano. It is a characterization is a very good important factor. That is it is the cost. The equipment is a very each and every characterization it is cost. Like BET, TEM analysis, it is a cost. Each and per sample 5,000 rupees, 5 to 6,000 rupees per sample. It is your preparation is good, no problem. Otherwise, you have suffered. Synthesis is easy, but analysis is very difficult.
Yeah. That is why I asked.
Yes, sir. Yes, sir.
You were telling about funding agencies, DST, Inspire, and all that. So
yes, sir.
you have to have some fellowship.
Got first
Otherwise,
rank in
it would
MSc
be very
Zoology.
difficult.
After I'm going to move on in my PhD in DSP Inspire Fellowship. Yes, sir.
Yeah. I saw your profile.
Yes, sir.
So that is why I asked you, because initially to get the samples analyzed,
Yes,
whereas
sir.
if we go for histopathology, or if we go for atomic absorption spectrophotometer, or if we go for any other analysis, it will not be that much costly.
Yes, sir.
HPLC or GC samples or any other samples.
Yes,
But
sir.
if the sample is related to nanotechnology research, then it costs a lot. So the preparatory part has to be very good.
Yes, sir. But the characterization is very good. It is a
Very
complication for the. It is cost analysis.
So nanotechnology is only for gold medals. Shekhar?
No, sir. No, sir. No. I'm not telling like this. Each
Because
and every person
it
synthesis of
involves a
nanoparticle.
lot of
Lot of research are working on nanotechnology.
nano research. Yes.
Yes, sir. Yes.
Any other questions from Zoom students? You can ask.
Yes, sir. Very good afternoon, sir.
Yes, Manasa. Speak loudly,
Sir?
Nana.
Yes, sir. Very good afternoon, sir. I'm Manasa Mitta from second year BSc Microbiology, sir.
Yes, ma'am.
As we are unaware of this topic of nanotechnology, but you have clearly explained, sir, and now we are easily able to get what is about the basics of nanotechnology, sir. Thank you for that.
Okay, ma.
Yeah, I have a question. May I ask you, sir?
Yes, ma. Yes. It's my pleasure.
Sir, as you told that zinc oxide and titanium dioxide and many other metal oxides, they have fungal and antibacterial properties like no, sir.
Yes.
When they are disposed into marine cultures like seas or oceans, does they cause any cell toxicity or does they affect marine organisms, sir?
Hello.
Yes, sir.
Please repeat your question. I can't hear you.
Now am I audible, sir?
Yes. Clear.
Sir, as you told that zinc oxide and titanium dioxide and many other metal oxides, they have antibacterial and antifungal properties, no, sir?
Yes.
After usage of those products, either cosmetics or any other products which they are made of, if they are disposed into marine cultures like seas and oceans, does they show any effect on marine organisms?
Yeah.
Does they cause cell toxicity?
Yeah. It is a good question. It is the ecotoxicity assessment. After we have used day by day, we have used for the some some lactic element, right? Lactic element. It is also ingredient of the major ingredient of the zinc oxide. It is a factor mainly involved in the concentration level. You particular in environmentally impact into the concentration. Up to the higher concentration, it is lead to the effect. Each and every organism have a different concentration toxicity level. So the in ingredients after the preparation or any material or any product, it is the particularly selected concentration. It is higher concentration, it is very leads to the toxicity. Definitely. It is a damage to the ecosystem. It's a planktons to it's animal, water, aquatic toxicology.
Okay, sir. Thank you, sir.
So we can also use nanotechnology in cell apoptosis.
Sir, there is another question
Yes,
from
ma'am.
Pushpalata.
Yes, ma'am.
How does the nanotechnology plays a vital role in development of a country, and is there any harm to human life? May I repeat the question, sir?
Yes, ma'am.
How does the nanotechnology plays a vital role in development of a country? And is there any harm to human life?
Hello.
Am
Is
I
there
audible,
any
sir?
I
is
can't
there
hear
any
you.
harmful effects to human life? One of the students wants to know. Nanotechnology, is it harmful for human life?
No, sir. Each and every nanoparticle not toxic to the human life because it is some chemical or toxic agent to the capping agent used for the it is a toxic in human life and ecosystem also. It is a nanoparticle preparation of some biologicals in phytochemical, some drug also not harmful in human life. In researcher, we have also used to further some doxorubicin in cancer drugs in combination with the drug into the incorporated the nanoparticle to treatment in the trials, in clinical trials. So the nanoparticle not harmful to the human life because of biosynthesis. We have used for the very toxic agent. It is very toxic.
Thank you, sir. There
Yes.
is another question from Swati Ramchandran.
Yes.
Sir, as they are used as nanoparticles are used in cosmetic products, will they
Yes.
cause any kind of allergic reactions to human
No.
skin?
No, ma'am. No, ma'am. Already we have used to for the some cosmetic items. Already I am already told in lactocalamine some creams used to for the zinc oxide, titanium dioxide. Majorly used to for the nanomaterial like two metal oxide nanoparticle used to for the cosmetics items. One is zinc oxide, another one titanium dioxide. It is a UV protecting agent, so they're not causing to the allergic some other things because the commercial to the product because tested for the in vitro and in vivo levels in the researchers. So the not harmful.
Thank you, sir. There is another question from Novira Sohali, ma'am. Sir, does nanoparticles are used in COVID-19 treatment?
Yes, ma'am. Already some researcher it's doing on the researcher some carbon C60 and quantum dots also preparation for the some nanomaterial used for inhibition of the coronavirus tested in the bioinformatics tools levels, but not fully not concentrated on the nanotechnology in the COVID-19. Maybe in future.
Thank you, sir.
Yes, ma'am.
Question
Ma'am, I have a question, ma'am.
Yes, yes. Carry on.
Good afternoon, sir. This is Pragna Chinmayi. As you have told that there is no problem with the human life. Is there any problem?
Yes.
Is there any toxicity in animal life or any plant life?
Sorry.
Uh, does these nanoparticles cause any toxicity in animal life? Uh, plant, not only the animal life, also the plants. Am I audible to you, sir?
Yeah, I told it is the depend upon the concentration. The nano material concentration level and used for the metals like zinc oxide and the titanium dioxide is not a harmful effect. It is used for the some lead, some another any other metal agent used for highly concentration. If it definitely not only the animal, the human also affect in the system. So we
Sir,
have
what
yes.
are the other metals that cause toxicity in every life? Like humans, plants, marine animals, everything, sir. What are the other metals,
Cadmium.
excluding zinc
Yeah,
and titanium
yeah.
oxides?
You repeat your question.
Excluding zinc and titanium oxides, what are the other metals which causes toxicity in lives?
There
Yeah,
are so many:
toxicity
cadmium,
material, right?
mercury, chromium. You can list. There will be so many metals which cause
Yes, sir.
toxicity to animals and
Yeah,
human
it is a
beings.
cadmium. Some of the cadmium, nickel. It is also highly toxic. Cadmium, nickel, and lead
Chromium,
also. It is
mercury.
highly toxic. Yes.
Can heavy metals be removed through nanotechnology? We are trying to study the effect of cadmium in crabs.
Yeah,
Since you belong to
you're trying.
zoology, I thought I would ask this question. You
Hello?
want me to repeat it?
Yes, sir. Yes.
Can cadmium metal toxicity be removed from crabs, freshwater crabs?
Cadmium, it is also a toxicity, a very toxic material, sir. It is
Yeah.
used for
If
yes.
If we induce cadmium toxicity to the crabs,
Yes.
to check what kind of toxic effect it is going to have, can we use nanoparticles to reverse the toxic effect?
Yes, sir. We have tried. You have tried for the some of the biopolymeric nanoparticle. Try for the toxicity removal. Like chitosan, it is one of the very good toxic removal biopolymer. You prepare for the chitosan nanoparticle to removal tested for the cadmium toxicity removal. I am already in my research. I am also used for the some zinc toxicity removal for the chitosan, bare chitosan. Is in used for the some Cereodapnia, microcrustacean plankton, used for the removal of the zinc toxicity. So you also used for the like chitosan biopolymer nanoparticle tested for the removal of cadmium toxicity. It's a very good effect, I think.
Thank you, sir. There is another question from Anand Mukherjee, sir. What is the role of nanotechnology in the medical sciences? Medical sciences, sir. Role of nanotechnology in the medical sciences.
Yes, ma'am. It is a drug delivery, majorly drug delivery application and nanotherapeutic application. It is a diagnosis and treatment. It is a main role. Some of the nano sensor used for the to diagnosis the disease and diagnosis the bacteria in food samples and water samples. So it is a diagnosis and treatment for the main role.
Thank you, sir.
Vijay, Bharat.
Bharat.
You can unmute and talk, Nana. Bharat.
Bharat, you could talk to the research person since you raised your hand.
Your voice is not audible, Bharat.
And also the final year students utilize this opportunity. Sir is giving so many so much information about how to proceed after your UG and PG. Please utilize this opportunity and clarify your doubts. Final year students. Bharat, unmute yourself and talk, Bharat. So if you have problem, you could text in the YouTube chat box so that we'll ask Bharat. Sir, there is another question from Arun Kumar, sir. What are nano clusters?
Nano clusters.
Yes, sir.
That's like nano clusters. It is a group of the particle. Like I'm already previously explained in my first slide. Like the metallic and the metal organic nanoparticles. It is a C60 and the carbon nanotubes. It is more over the all particles. It's aggregation, agglomeration of the particles. It's like nano clusters.
Another question from Mahesh, sir. What is the key role of nanotechnology in aeronautical engineering?
I don't know idea, ma'am. Any key role in aeronautical engineering? I think any nano chips and nano sensor. I think it is the main role in the aeronautical engineering. It's a nano sensor devices.
Thank you, sir. Over to Pragna.
Thank you, ma'am. Thank you, sir. I request our head, Department of Biological Sciences, Dr. S.V. Ravikant, sir, to propose word of thanks.
Thank you, Pragya. On behalf of the organizing committee, I profusely thank Dr. Shekhar Vijay Kumar. Dr. Shekhar is a gold medalist in zoology. I also did my PhD in zoology.
Oh,
So
thank
I
you.
know how tough it is to work in that, and how enthusiastic one would be to read about various aspects of zoology. Nano research is one of the hotcakes, and I am sure students who embark this area are sure to settle down in their lives. I am sure today's resource person delivered the talk to the fullest satisfaction of the students. I am particularly impressed with the way he very patiently answered all the questions of the all the questions that were posed by the students and other participants. I am sure the participants are benefited with knowledge they gained, and this would help them to settle down comfortably in their lives. I would like to thank the alumni of Sri Vidya Niketan. One of the alumnus, he mentioned that he was also working in nanotechnology. Can I also join today's webinar? He asked me, Mr. Karthik, and I told him, yes, you can also listen. So I'm thankful to the alumni of Sri Vidya Niketan Degree College. My sincere thanks to our beloved and dynamic principal, Dr. Asha Lata Madam, for all her support and encouragement. And I thank our UG and PG coordinator, Dr. P. Manohar, for his support. I would like to thank the faculties of computer science for all the technical help in organizing the program. I also take this opportunity to thank the media team for designing the e-brochures and certificates. I would like to express my heartfelt thanks to all the participants and students without whom this event would not have been unfolded. I thank student volunteers, Ms. Pragya and Mr. Tejesh, who anchored today's webinar. Thank you, Pragya and Tejesh. I thank our management for supporting us in all possible ways in making this event possible. I would like to thank Dr. Charles Lekhya Priya for taking the responsibility of organizing today's webinar. She coordinated with all and made this program possible. As usual, I would once again like to repeat this statement. You are a huge asset to the Department of Biological Sciences in particular, and SVDC in general. Thanks, Amma. I also thank Dr. Sridevi for all the help she lent in organizing the webinar by taking the YouTube question and answers session. Thank you, one and all. And back to you, Pragya.
Thank you, sir. I thank all the participants for joining us today. As you can see, the feedback link is given in the YouTube chat box. Kindly fill the feedback form to receive an e-certificate. E-certificates will be issued on or before 28 September 2020. Thank you one and all. Over to you, Dr. Charles Lekhya Priya, ma'am.
I would like to add a few words here, ma'am.
Yeah, ma'am,
Because
sure.
I was really impressed with the presentation of Dr. Vijay. Dr. Vijay, it was really great listening to your presentation, and I was trying to understand each and every slide where you were trying to be very very clear about how to go about the research of the nanoparticles or the biomedical or the nanomedical applications. I think our students can take immense value from this. Now, I see as a teacher or as a mother, I see that you are the personification of positive research attitude where you are trying to explore whatever is possible to do research in the areas which are very much useful for the existence of the human civilization. So I congratulate you on this occasion, and I congratulate your university also for taking very good initiatives in the research areas. So this is a special thanks from all of us here from Sri Vidya Niketan Degree College. And I hope that with this inspiration that you have given, I'll also start a little bit of research in the metallic oxides present in a lot of spices as you were discussing about, and see how best we can make our cooking also very exceptionally healthy, to make nanomedical applications in the cooking also and make our children healthy in the coming days. Thank you, Dr. Vijay, for being with us
Thank you,
here
ma'am.
today.
Thank you, ma'am. Thank you, ma'am.
Thank you so much. Thank you.
Thank you, ma'am.
you, ma'am. On behalf of Department of Biological Sciences, I am very happy to express my sincere gratitude to our Honorable Chairman, Dr. M. Mohan Babu Sir, our revered CEO, Mr. Vishnu Manchu Sir, for giving us an opportunity to organize this event. I am very grateful to our principal, Dr. D. Ashalata ma'am, for providing guidance and encouragement. Thank you, ma'am. I would like to thank our head, Department of Biological Sciences, Dr. S.V. Ravikant sir, and our UG and PG coordinator, Dr. P. Manohar sir, for their constant support. Thank you, sir. We are extremely thankful to our resource person, Dr. Vijay Kumar sir, for accepting our invitation in his busy schedule and sharing his valuable thoughts and experiences with all of us. Thank you very much, sir.
Thank you, ma'am.
We have participants from all over the globe. We thank all the participants without whom the event wouldn't be successful. Thank you, one and all. As no program can become successful with a single person, so I extend my big thanks to the organizing committee members, Dr. V. Sridevi ma'am, Mr. K. Munishankar sir, and all our student volunteers, Ms. PVN Pragna Chinmayi, Miss AP Vaishnavi, Mr. T. Tejesh, and Mr. Pranava Upparlapalli. Thank you for making this event successful with your contribution. Feedback link is displayed in YouTube chat box. Those participants who fill and submit the feedback receive certificate on or before 28th September 2020. I repeat, feedback link is displayed in YouTube chat box. Those participants who fill and submit the feedback receive e-certificate on or before 28th September 2020. Take care, be safe. Thank you, one and all. Principal ma'am?
Yeah, ma, we can leave. Yeah, yeah. Thank you. Thank you, one and all. Take care, everybody.
Thank
May I
you,
close
ma'am.
the session, ma'am?
Yeah, yeah. Please, please. Thank you.
Thank you. Vijay sir, thank you so much.
Thank you, ma'am. Thank you, ma'am.
And Principal ma'am, thank you. And all other committee members, thank you, one and all. Ravikant sir, thank you. Manohar sir, thank you so much. With this, we
Thank
are
you,
ending
ma'am.
the session, ma'am. Shall we end the session?
Please, please. Thank you, everybody. Take care. God bless.